surprise, I guess, spent a lot of my research career developing genetic modification technologies and applying them in the chicken. And I apply them across the range from the chicken as a developmental biology model, through biotechnology, making valuable proteins in the eggs of transgenic hens, through to animal breeding. But today I want to talk about genome modification more generally in farmed animals. Some of the things I'm touching on have already been touched on, I always, if I'm talking about genome editing in farmed animals, particularly in chickens, I always spend a little time at the beginning introducing breeding and the achievements of animal breeding, because I think these are really not understood. And if we're doing genome editing, if we're applying genome editing, we're applying it in the context of very effective animal breeding that's been going on really since the Second World War. And an example I like to use is the change in the broiler, the meat-type chicken, from 1950 to now. It's particularly, as mentioned earlier, for layers, they've become much more feed efficient. You can now get 590 kilograms of meat per ton of feed, whereas it was only about 330 per ton of feed, close to 80% improvement. Again, that reflects also back in how much land you need to produce that feed. So animal breeding is very efficient, but why do we need new technologies still? Why do we need, for example, genome editing that I'm going to be talking about? And this is a slide from the Bill and Melinda Gates Foundation, and it predicts that the world demand for animal protein is going to go up. And although we talk in Europe and the US about becoming vegetarians or vegans or eating less meat, really it's the rest of the world where that consumption is going to go up. So we can make our changes, but they will not have much impact on this predicted increase. And in particular, the increase in meat from poultry. It is from 82 million tonnes up to 181 million tonnes between 2005 and 2050, and in eggs from 62 million to 102 million tonnes. So these are enormous increases that are being predicted to be required. So how are we going to deal with this? Well, we'll hear a lot more, and we've already heard about using genome sequence for breeding. And the sequence of the genomes of all the major farmed animal species are available, and the knowledge and the information is getting richer by the minute. But this gives us information which could also be used if we're going to use molecular techniques in breeding, like genome editing, as well as in improving our selection technologies. And we can use these because we already have developed technologies for genetic modification of farmed animals. So we know how to manipulate, this is a pig egg being injected with a transgene, and here a pig expressing green fluorescent protein, and these are chickens expressing green fluorescent protein. So we've added new genes with completely new characteristic to these species. These are both from the Roslin Institute. But genome editing is the latest sophistication in genetic modification. And just in case not everybody here is up to date with what genome editing is, basically genome editing allows us to go in to DNA, so this is a chromosome that's unwound, and specifically identify a single nucleotide in a specific gene to edit, to make a genetic change. And this is an illustration that many of us use. We make that genetic change by using molecular scissors. So it's an enzyme that we can send in with a target to a particular sequence and allow us to cut the DNA at a very specific sequence of a gene that we have chosen. And we can cut it, or we can cut it and we can replace it with another sequence. So we can make very specific genetic changes that we have designed before we made any of these manipulations. And we can do this because molecular biologists over the last decade have been developing novel enzymes that are sequence-specific, and they can be targeted to a specific sequence. And the most recent one of these is CRISPR-Cas9, and that's all over the news now, particularly because it's being used in human medicine. And CRISPR is an enzyme called Cas9, which will bind to DNA and it will bind at a specific site if you give it a small RNA molecule that is complementary to the sequence of your gene. So you can target your enzyme to a gene, and then the enzyme will cut the DNA. And it's very, very efficient at finding this target sequence. It's absolutely remarkable, and I still don't quite believe it. And if you cut DNA, it will repair itself, and that can cause a novel mutation. Or you can cut DNA, and I think the next slide shows this better. You can make a specific change. So if you make a break in DNA, your cells are tooled up with enzymes to repair it. For example, if we get UV, we cause DNA breakage, and we need to repair it. And that repair can be called non-homologous end joining, which is basically sticking the broken ends together, which can cause a single nucleotide change, but more often cuts a few out or sticks a few extra in. But it's at your site, your chosen site. Or if you cut the DNA and you have what we call a donor template, which is homologous to this one, but has a sequence in the middle that is either completely different or just different by a single amino acid, for example, in a protein, you can get that donor template to be recombined into the cut, and then you've introduced a precise insertion or modification. So this is what genome editing is. So it's making, and it's exactly the same technology being used in plants and animals. So going on to animals, what are the potential in farmed animals? Well, I think you can break it down into these 4 different things. We can think about moving beneficial gene alleles between breeds. We can move beneficial gene alleles between species. We can improve characteristics that are really challenging for genetic selection, for example, disease resistance. And we can maybe generally accelerate genetic selection. So the rest of my talk, a lot of it is examples about what's going on in animal breeding. And the first one is about moving an allele or a mutation between breeds. And in cattle, beef cattle are genetically hornless, and dairy cattle on the main have horns. And this is just something that I don't quite know why it happened, but it happened a long time ago, that beef cattle were selected from cows that had a mutation that caused them to be genetically hornless, but dairy cattle have horns. And it's a welfare issue and a cost issue, because in most dairy cattle, the horns are burnt off in calves, so they're physically dehorned. And what this company, Recombinetics in the US, and now working with people at UC Davis have done is the mutation that caused the hornless cattle was identified and the sequence known, and they have introduced it into dairy cattle. So this is one of their first genetically hornless dairy cows. And Recombinetics are working now, I think, in Canada to take this mutation into dairy cattle. So it was obviously chosen, I think, as a first go because it is a very clear welfare benefit. My next example is from colleagues at the Roslin Institute, so that's Alan Archibald, Bruce Whitelaw, and Christine Tate-Burkhart. So they're working in pigs, and the disease PRRS, porcine reproductive and respiratory disease virus, is a really nasty disease of pigs that is spread across the world. The data that I have from them is that it costs $650 million a year to the US pig industry. The UK pig industry, 145 million odd per 500 sow breeder finisher herd if they're not vaccinated. And the vaccine is not particularly protective, it's not very good. But a lot is known about the biology of the virus and its infection. So this slide here shows the cell membrane of a pig cell and the virus coming in. And the virus has a spike protein complex on its coat that interacts with this protein CD163 that's on the cell surface. And this is absolutely required for the virus to infect the cell. And it's known that the virus, it's a chain of globular domains, the protein. And it's known that some parts of it have a function, for example, binding to hemoglobin. And it's known that this globular domain number 5 is the bit that the virus binds to. Whoops. my colleague Simon Lillico suggested that you could snip out this domain in the gene, and then the virus would not be able to infect. So they did that. They designed this globular protein domain is encoded. This is the CD163 gene. It's encoded in one exon of the gene, and they designed CRISPR, and they're targeting RNAs to flank exon 7. And they showed that they could cut out exon 7 of this CD163 gene, and they generated pigs with this mutation. So these are wild-type pigs, heterozygous pigs, and mutant pigs. And the first thing they did was take cells from those pigs and see if they could infect them with the virus. There are two big major strains of virus, and they used both strains and did the same experiment. And you can see here that the wild type and the heterozygous pigs died, the cells died, and the mutated cells seemed to survive. So once they'd done that, they felt that they were justified in going on to an animal experiment, and they took 4 wild type and 4 mutant pigs, and you can see here, you can't see any difference between the pigs. And they put them in a pen together and at 7 to 8 weeks of age, and they infected them with the PRRS virus, and they co-housed them to allow natural spread of the virus as well. And basically the answer is that the wild-type pigs succumb to the disease, and the gene-edited pigs show no sign of infection whatsoever. So Christine is taking this further to examine the robustness of these pigs, their livability, their response to other infections, because you know, there may be unintended consequences of making this mutation. My next 2 examples are things that are just on the way, but that's because we're early days with this technology. And this is a project that Bruce Whitelaw at Roslin and his colleagues are doing, and this is to do with African swine fever virus, which was in Africa but is now moved into Russia and is coming through Eastern Europe, heading this way. And it's at least partially dispersed by ticks. And it's known that ticks will the ticks will bite warthogs, that's the wild pig species, and they get the virus, but they don't get sick, whereas our Eurasian pigs get very sick. It's a major problem. Bruce's group have identified an allelic difference in an important protein that is involved in the virus infection between these two, and they're currently have made pigs that have the warthog allele of this gene, and will be shortly doing an infection study with African swine fever virus. But the reason I use this example is that it's something that we could not achieve by crossing. You can't cross a warthog with a Eurasian pig to transfer beneficial genes or alleles. And I think my last example is from my colleague Ross Houston, and this is to highlight the potential of genome editing in aquaculture, because aquacultural production of fish and other species is really increasing, and genetic improvement has not been implemented nearly for so long in these species like salmon. And they identified, looking at this nasty virus, infectious pancreatic necrosis virus, that there's a genetic variation in resistance to IPNB. And they showed that there was a very effective resistance and identified resistance versus susceptible locus. And they've mapped that down to a single chromosome. And we've now set up the technology to do genome editing, and they're trying to identify the allelic difference underlying this. And I think this will relate back to what Bruce is talking about, but if you can identify the allele, you can use editing to move it, and rather than doing the domestication drag, that I've now learned is the term I should be using, of crossing to introduce that beneficial allele, where you may be making other changes that you really don't want to make during breeding. And my final example is a theoretical one. So this is my colleague John Hickey suggests that if we could use genome editing to introduce many allelic changes, we could accelerate our rate of genetic gain. And he's modelled here going from 1 to 20 edits and predicting the increased rate of genetic gain. But of course there's a problem that we really need more on the animal physiology side to get together with the people on the genetics side to work out how we identify these beneficial alleles. And I think that's really one of our big challenges, especially if they're alleles that have a relatively modest benefit. So I hope I've given you some examples to show you that there is potential for genome editing and we can do it. But there are really, this is where I've realized relatively late in life that you can't just sit there doing your work in the lab. We have to think about how we get how we are allowed, how we get the ability to apply these improvements. So there are a lot of questions. Is it GM or is it not? That is genome editing. Is it the same as GM? Is it not? Where does genome editing end and GM begin? And regulation, there's regulation in Europe, there's regulation in USA, in China, in South America. The regulations in all these different countries are different. We're told that we may have a benefit of Brexit, in the UK we'll be able to have our own regulations, but that would be of no benefit because we wouldn't be able to sell our produce in Europe. So, this is something that really needs to be tackled at an international level. There's the issue of labelling. How do we label these? In Europe, UK, we label things as GM. Well, that's completely meaningless to my mind because you've got to say what the modification is. And if it's a genome edited allele, you've made a single nucleotide change. How do you label that? And I know in the US, things get a little butterfly to say they're not GM, but now there's a move to label things to say this contains GM ingredients so that you stop this emotional business of highlighting something as good or bad. And I think amongst those involved in genome editing and genome engineering, there's a strong view that we should switch to regulating the product, not the process. But where are we in animals? So I think it was mentioned earlier, there is one GM animal that has been licensed for human consumption, and that's the aqua bounty salmon. And that was licensed in 2015, but as far as I know, it's for sale now in Canada, but it's not for sale in the US, unless I'm behind the times on that. I believe the reason is that it's not clear how it should be labeled in the US. So although they are licensed to sell it, how they sell it is not agreed. And actually, the aqua bounty salmon is something that has been an ongoing story for a very long time. So it was one of the first GM farmed animals where they express growth hormone through the winter so that the fish are basically carry on growing through the winter and are much more feed efficient. And they made this in 1989, put in their first application to the FDA, who regulate GM animals, because they are seen as an animal drug. In 1995, their application wasn't complete till 2009 because of all the hoops they had to go through. In 2010, the FDA said it was safe. They did a draft environmental risk assessment a couple of years later, and it wasn't approved until 2015. So it took an enormous length of time, and people keep saying this will happen again. Well, I think we have to, things have to change, or this technology won't be applied. There are some lights on the horizon. The US government has approved transgenic chicken, but actually this is one that makes a valuable protein drug as a component of egg whites. So it's from a genetically modified chicken. It's something I find a lot is, it's the purpose of your activity that sways people. If it's for a human health benefit, people will be much more convinced than if it's for a multinational company to benefit. The pig breeding company Genus is on board with the PRRS resistance, and they're taking that on in the States, and they're talking to the regulators in the States. So they're a breeding company that have put it out there that they are considering using genome editing. I haven't talked about what I do, so my interest in terms of animal breeding in poultry and using genome editing is in flu resistance. And we have several projects at Roslin going on where we might use genome editing. One is using genetics to look at birds that survive flu infection and comparing their genotypes with birds that don't. There's a lot to do with understanding the biology of flu infection and can we find targets to edit. And so I realized that we need to be upfront about what we do. And this is me looking slightly terrified because I'm talking about why do we need GM chickens to an audience of 1,500 people at a TEDx meeting in Glasgow. And I have to say that I find that if you go out and talk about this, I've never had any real trouble. People are prepared to consider the benefits, the risks and benefits of using genetic engineering technologies. But of course, I use the example of flu resistance because you can show that there will be clear benefits to the chickens. There's potential of human health benefits as well. It's something people know about already. So it's, to my mind, a good example to be working on. But also, there have been a couple of studies with the UK public come out this year. So the New Scientist magazine runs a survey, Ask the Public, and this is from this year, and they asked about genetic engineering. So in general, the public don't distinguish between genome editing and GM genetic engineering. I try and use the term genome engineering as much as I can remember to. And they were asked, would they approve of genetic engineering if it could cure or eradicate diseases? This means humans, 80% said yes. If it could help us to produce better crops and livestock, 47%. And slightly to my mind scarily, it could be used to improve human capacities such as intelligence, 45%. that one I find scary. But I haven't got a slide from it because I failed to extract a graph. The Royal Society in the UK put a lot of money into a public dialogue and survey on GM technologies, and they had groups looking at human, looking at animal agriculture, and looking at crop science. And on the whole, about the highest number of people thought it was great for human health, the next for plant breeding, and the last was animal breeding. But of course, that's where the welfare issue comes in. But even for animal breeding, people are very willing to consider it and approve or possibly approve is around half of the people that they surveyed. So I was very impressed with this Royal Society one. I put the link up here because there's a big document come out of this survey. It was done in great depth. That people were prepared to engage and consider the risks and benefits. And if they are for the benefit of mankind, people are generally much more supportive. So I think I'm running out of time. So I would like to acknowledge my colleagues, Bruce Whitelaw. That's not Simon Lillico, the pig, Simon's person. Do a lot of the work in pigs. Christine Tate Burkhardt and Alan Archibald, who Christine has really taken on the PRRS project. She's a virologist, so it really shows that it's very important to have people who understand both the biology of the disease and the genetics, which is provided by Alan. Ross Houston and John Pavlin, who are carrying out the work in salmon, and this is Ross running for the marathon for Scotland, and John Hickey, who is our genomics and breeding whiz. And we had a visit from Bill Gates earlier this year, and he said a lot of things, and they've made very useful quotes. So he is really on board the use of genome engineering, genome editing, and he said never before could we talk about making gene changes that can make a species completely resistant to disease. And I think with that optimistic view, look for questions.
Yeah, thank you very much. Are there any questions?
Or comments?
Don't be shy. So if not, I would have a question, so maybe so that people can hear me. Maybe it's a bit special question. You were talking about bird flu and the resistance to bird flu. So is there the risk that if you breed birds that do not signs of infection, that they can still spread the virus, especially with the flu? Yeah. It's a bit with the antigenic drift and shift.
Yes.
And
yes, absolutely.
Is that a problem?
So you know, I think we're really at the beginning of this, and one of the reasons we're really at the beginning of it is that much less research has gone on because people think it will not be applied. And we need to move on to modelling and experimentally determining those risks. So with the PRRS, they're trying to replicate PRRS virus on these cells that can't be infected. And that, to my mind, is a really nice example because the virus never gets in. It doesn't get a chance to even replicate a little bit and then select for something that overcomes that resistance. So we absolutely have to be very careful. And also we have to think about the model of using antibiotics. You know, if we use genetic resistance, we can't think that's the problem solved. We have to think about, if we have genetic resistance, do we rotate the resistance so that we don't promote evolution of the virus?